So welcome everybody to a truly exciting edition of our online seminar series on emerging topics in biomolecular magnetic resonance. Please don't record, because both speakers of today agreed to record the lectures, and we, or actually Kevin Gardner, will upload the lectures then in due time to the ICMRBS YouTube channel. Just to remind everybody that we don't use the chat, but do use the Q&A box for putting in your question. And of course, it's always very useful if you already do that during the talk. Or you might also raise your hand and then we can unmute you and you can ask the question directly to the speaker. I also remind you of the early career online seminar series of ICMRBS, which I guess is very successful. And please don't forget that next Monday, the ENC starts, and our Palmer just told me that there's still time to register. So a great opportunity, of course. Without further ado, I now hand over to Claudio Lucinat, who will introduce our first speaker, Lucia Banci from University of Florence.
Okay, thank you, Marcus. It's a pleasure to introduce Lucia. I know that most people in the audience probably know her, but nevertheless, I'll say a few words, not to steal too much time from her lecture. So Lucia is director of CERM. As you know, CERM is the Magnetic Resonance Center of the University of Florence. And, it's, besides being our magnetic resonance infrastructure, it's also part of INSTRUCT, which is Integrated Structural Biology. And the CERM is the Italian Center of the Integrated Structural Biology Infrastructure, INSTRUCT. About Lucia, I think she has been from the very beginning involved in the birth and development of INSTRUCT. She's been one of the, well, inspiring figures or promoters, whatever. And she's presently member of the INSTRUCT Council, member of the Executive Committee of INSTRUCT, and of course the coordinator of the Italian Center, which is an NMR center. And I would like to underline this because Lucia has been always acting as a promoter, or I should say defender of the role of NMR in INSTRUCT, and I think up to now successfully, I would say. In terms of science, you know that Lucia started NMR very early in her career, and the theoretical side, especially the electron nucleus interaction, which is a kind of a specialty of our place, and she has been fostering that for many years. And then she moved to structural biology, and that justifies her involvement in INSTRUCT, of course, and more lately in cell NMR, which is well, a promising technique. And this is also the topic of the talk of Lucia today, which is in cell NMR, a powerful approach for drug and fragment screening. So please, Lucia.
Thanks a lot, Claudio, and thanks to you for inviting me to present these results and for organizing this series that in this particular period of our life is very, very useful to not only maintain contacts, but to be just exchange experience and exchange results and comment and discuss on this. And what I thought it could be useful to present is some of my most recent applications of in-cell NMR, specifically devoted to drug and fragment screening. And I have been doing together with Enrico Lucinat, Letizia, Matteo, the development of in-cell NMR, in particular, exploiting the idea of overexpressing proteins directly in human cells, and having directly expressed the protein inside the cell instead of inserting them while produced by bacteria, it allowed us to characterize a number of processes like maturations and folding since the beginning, because the protein are directly produced in the cell. And this is already data in the literature already since a number of years, and there is a number of review also with Enrico Lucinat and myself, so this is something covered. One of the potentialities of in-cell NMR is indeed characterizing some functional aspect inside the cell. And the one point that is relevant is, for example, just I take an example of this, is monitoring the cysteine oxidation state that we characterize for a number of proteins, which they are present in cytoplasm, and everybody thinks that being in the cytoplasm, this protein must be in the reduced state. And this is not true, because the reduced state of the protein is a very tightly controlled process and depends on the presence of other proteins which maintain the protein either in the oxidized or in the reduced state. Those are also data are in the literature. I want just to present this specific example where the oxidation states we can characterize not only as it present when we produce the protein in the cell, but also we can monitor the effect of oxidative stress. And so then, for example, this protein, which is a dimeric protein and involved in a series of pathological state conditions, this protein is sensitive to oxidation when is undergoing cellular stress. Indeed, if we treat the cells with hydrogen peroxide, we induce oxidation. This was already reported, but what specifically happened in the cell was not clear. And then we were able to identify, using directly nitrogen 15 labeled cysteines. This is on the lysate, we can monitor in the cell. We obtained, we clearly show that there is one specific cysteine. Having the assignment, we can tell is a specific cysteine that get oxidized, and isolating this protein from the cell, we can definitely clearly show that there is form SO2 species and not SO or SO3. That was also another debate. So this is just an example of what one of the many things that in cell NMR can address. And regarding the oxidation of cysteines is also relevant for interaction with the small molecules, and some of them can be treated as drug or potential drug. And in the case of SOD, which is undergoing aggregation in neurological disease, amyotrophic lateral sclerosis, has been found a large amount of aggregates, amorphous aggregate of these proteins. It had been shown that if one free cysteine is stabilized, then this prevents protein aggregation, because the protein aggregation in this protein occur through when the protein is in the immature state and has not a metal ion bound there, but also when a disulfide bond is not forming an inter-protein disulfide bond, but also when these cysteines, which are reduced and freely exposed the solvent, can get oxidized, forming interdomain disulfide bonds. So this is one strategy has been shown by a number of labs, also early days by ourselves. But what we also, with this spirit of somehow blocking the cysteine, there was the analysis of the behavior of empselene, that is already used drug, is a selenium-based drug that has been already is used as anti-inflammatory and antioxidant activities. And what we found that this drug indeed stabilized the structure, even the mature form of SOD, because it coordinates to this cysteine. And then with this stacking interactions, stabilize these two subunits, even if are not totally fully formed. The point is that this is what is happened in vitro. Then we analyze, the point is that this binding is relatively labile, or can be broken by reducing agents. So with treatment with DDT and glutathione that is present in the cell that break this type of bond. Anyhow, that is a possibility to use this as drug, but then we check this through in-cell NMR, and the behavior in the cell of this potential drug for preventing SOD aggregation is totally different. And indeed, what we found is that this drug promotes the formation of the intra subunit disulfide bond. In this way, stabilizing the protein. So it stabilizes the protein and prevent aggregation through a totally different process. And in particular, we can see that, for example, this is one ALS-related mutant. This, without the drug, this protein never get properly folded and the mature state with the correct disulfide, both physiological disulfide bond and binding of metal ions, while in presence of this, it acquired a complete folded state, which when it's complete mature state, it doesn't aggregate. So this gives a message regarding the role of this drug in the cell, but also it tells the information of how vital the analysis in cell is essential, because you have a totally different behavior and effect if you characterize in vitro with respect to in cell. And then going on regarding the interaction of target proteins with drugs or potential drugs, then in the most recent time, something like 1 or 2 years or so, we tested this approach, I mean, the cell NMR for screening, for monitoring the effect and the behavior of already existing drug or potential drugs against targets present in the cytoplasm. And for testing this, so we tested the number of targets, and we, I'll show you the results, but then I can tell that this can be a very useful and approach, because in cell NMR can address some points and can monitor the efficacy of a drug or lead and the relative initial steps in the drug development pipelines. And what we tested this approach and developed this approach against human carbonic anhydrase 2, which is an enzyme which catalyzes the transformation of CO2 to hydrogen carbonate. And this is isoenzyme 2. We have 16 isoenzyme isoform, and this is present in the cytoplasm. So we express this protein in the cytoplasm of X cells, so human cells, and we monitor the effect of ligand binding. We can see that, for example, this is just the classical HMQC. We can see some specific spectral changes when this is treated with this is already an approved drug, is already used drug against the glaucoma, and this is you can buy in the pharmacy shop. So the potential, the real powerful of this specific target is that the active site, and so then the inhibition site, is a zinc ion, which is coordinated by 3 histidines. And this, the NHs of the 3 histidines are relatively shifted outside the magnetic envelope. This is the magnetic, there is a zinc here. This is the native state of the protein. And so then we can monitor the binding of the inhibitor directly from 1D NMR spectra. These are 1D NMR spectra collected on cells, living human cells. So we don't even need the labeling. So in this specific case, otherwise with just a simple uniform nitrogen 15 label, we can follow the the screening. And in this specific case, what we learn? Again, we learn that the behavior in vitro with respect to the cell is quite different. So if you look at this is the control experiment, these are two already existing validated drugs, and there you see that there are spectral changes indicating binds and efficiently bind. But then look at molecule 1 and molecule 2, for example. These two molecules, they have exactly the same affinity constant, and molecule 1 binds, molecule 2 doesn't bind. But if you do the same type of screening in vitro, you get exactly both of them, they bind with the same affinity. So the behavior is totally different if you analyze this in vitro and in the cell. And this is, as I said, a very important point because it determines a checkpoint while developing drugs, molecules to be transformed as drug. And what we did also analysis as a dose dependence response, the binding and time dependence, and we found that what is really limiting the rate limiting step is the diffusion through plasma membrane, and we can from the fitting of this and other data we can determine the coefficient, the membrane permeability of each molecule, and we see that even these even already approved drugs, they have a different membrane permeability, and this is correlate with perfectly with the potency of the drug. So one of the two drugs is required 10 times more as quantity for treating glaucoma, and it has the permeability coefficient 10 times less. So that there is really a striking correlation between this efficacy and the pharmacological relevant dose. We analyze also the behavior of a number of other approved drugs, other drugs. Some of them were approved not for carbonic anhydrase, but for other targets. So here is the list of these. And some were other inhibitors for carbonic anhydrase, and then for other type of diseases, and some for a different isoforms. These are for calcium channel or other transport. So the number of molecules, and we found in vitro all these molecules are inhibitors of carbonic anhydrase. So if you do the analysis in vitro, would be happy with any of these molecules. But then if you look at in the cell, we see that some of these molecules perfectly bind, other partially binds, and you see that there are multiple forms, because some of them are, this is the sum, and we can convolute the spectra, and these are the sum of the fully bound form that we can obtain in vitro and the unbound form. And then we see that the fraction that even if with a very high level of inhibitors, there is in some case incomplete binding. And we can classify different behaviors. So then there are some molecules that these three molecules, so they are faster diffusing, and then okay, that are no difference in, with respect to behavior in vitro. Then there are other that are slow diffusing, and so require a lot of time, and so the amount of bound fraction increases over time. So then this here is a matter to be considered when you develop this for drug with respect to pharmacokinetic analysis. And then there are some other molecules that are peculiar because are unstable binding. So then we see that initially we have a large share of protein bound form, but then over time, this ligand is released. And so then here, this is a matter of obviously to understand further, which is the cellular process which induced this. But it's very important to understand that this target, despite binding with the other affinity, then at some stage there is the release of those specific molecules. To go on and characterize further the behavior of this drug over time and with real time, because what I show on as a function of time was the function of incubation time of the cell with this ligand, but to have real time, we implemented with a number of people at CHERM a kind of bioreactor based on the Bruker Insight setup. And yeah, this is based just in a few words. This is already also published. There is a membrane which is able to, where we flow the nutrient and obviously oxygen. So external, this is the NMR tube. Here there are the cell, and through this membrane, then there is this flow, and then it's coming out the bioproduct. So then cells are continually supplied with the nutrient and oxygen, and so then they remain alive for quite a long period of time. And indeed, for example, just if you check, we analyze the phosphorus 31 ATP production, and immediately as the flow is switched off, then the cell die. That is a very clear correlation, and in some cases on some differences which occur when the cell are preserved or the cell are dead. There are reshuffling, for example, of these metal ions in SOD. This was analysis on SOD. So these are still alive and these are dead. And so this is when the flow is off, the cell die. And we were able to preserve this as long as 40 hours. And we exploited this, and just the last example to show this ligand binding by real time, exploiting this bioreactor. We started from this again back to carbonic anhydrase, and we flow inside this two already existing drug that have, however, different diffusion membrane permeability. And then you can clearly see that over time, this is a very fast changing and uptake, so that this is the unbound protein and this is the spectrum of the bound protein, while for this other drug, it takes much more to evolve in this scale is hours to reach the fully bound form. So then you see that these are clear spectral changes. So this is fully bound, and this is the control, so the free target protein. And so then it can be through a analysis of this spectra, we can extract the information of the fraction of the bound species protein with respect to the free one, and then we can monitor the kinetics of the uptake and binding of this protein. So then you see that this how is much faster this one with respect to this other drug. So this is a very important information as well, while developing new drugs. And then this type of characterization can be easily done at the very, very early stage, before a lot of chemistry is done and a lot of implementation, of where you go to more cellular and organism analysis and characterization. So this is more or less what I want to say, and this is just a picture of CHERM, where there are all these various NMR instruments, the EPR, we have a EMR biobanking, the relaxometer. So this is what most of you probably know, but we have really the largest magnetic field range because we can measure some NMR parameter 0.01 megahertz up to 1.2 gigahertz. And this is the picture of the new instruments. And I want to thank you, and particularly all the INCEL NMR group people and you for attention. 20 minutes, Lucia. I think.
Should I, there are 3 questions already.
Okay.
Should I go ahead with that?
Yes.
Oh, the first 2 questions are from John Tainer. First one is exciting work on comparing in vitro and in-cell results on SOD and so on. For the in-cell drug screening, what is needed for good signal to noise and other challenges to getting target-specific results? And then related to this, the second question is, would the current technology allow NMR drug screening with organoids instead of cells?
Starting from the last one, yes. We have, for example, characterize and collect in organello NMR, for example, proteins that we were inserted through the cell with a specific targeting peptide in mitochondria, and then we isolated mitochondria and we collect the spectra on that. So this is, as long as you have the protein inside there, that is the target there is possible. Regarding which are the limitations, first of all, if we work on looking at protein detection NMR, the main limitation is the detectability of the protein. And this beside the size, because there is the classical NMR size, which is even a little bit lower in the cell, but not that much. But the main problem is the detection because a target protein might weakly interact with other proteins, with large assembly, with the membrane. And so that in that case, it became invisible because it doesn't tumble fast to get the NMR signal. And so then in that case, we have shown for a number of other proteins, not as drug target, but proteins, having an idea about which are the interaction, we can design specific mutation far from the active site, and so then we recover the NMR signal. So that is one first critical point if you want to do the analysis as a target monitoring. If you do ligand monitoring, then you need some type of labeling, for example. There are examples in the literature by a number of groups looking, for example, fluorine 19 drugs or small molecules to report regarding the binding. In terms of sensitivity, it really depends on the quality of the spectra. We have observed in cell NMR spectra as low as something like 30 or 40 micromolar concentration, 30 micromolar concentration.
We have another question related to this from Walt Chasing. Great concept, beautiful idea, for example, to directly test for off-target binding of a potential drug. Now the question is, overexpression of transfected proteins in cells create artificially high levels of protein. Are these concerns about how effectively this relates to physiological conditions where levels are substantially lower?
That is obvious. This is true. I mean, we increase the level of the protein in order to detect them. And this is something which can be, for some aspect, that can be addressed if we can insert label protein in heterologous produce. But still, there will be a little bit of background, and still there is some limitation. If the target is very low, if it's a nanomolar in the cell, still is not possible by NMR to detect the target. Still, in some extent, as initial screening is still very, very powerful because you monitor whether the the drug or the molecule enters the cell or not. So this is a key point, because while if it enters, it diffuses to the target, this is a key information that you can have very early in the stage of analysis of the drug development.
Okay, Lucia, we have 3 more questions. Try to provide quick answers if you can. Yeah. So one from Guy Montiglione, beautiful presentation. Do you have any indication that some drugs may bind targets with apparent KDs that are tighter inside the cell compared to what is measured in the test tube?
Not in my experience, and not as a sorry, but I don't have that experience.
That would be interesting.
Yes.
Another question from Nicola Salvi. Thank you for this exciting talk. Do you know how much of the effect of the kinetics of ligand binding in cell can be reproduced using viscogens in vitro?
This is a very good point. Sorry, I have to elaborate. I never thought about that, so I cannot give a quick answer. I have to elaborate. I don't know. Sorry.
The last question is from Himanshu Singh. Great talk, Lucia. Did you observe any PTMs in your in-cell studies of carbonic anhydrase 2?
What is PTM?
I'm not sure I know what a PTM is. Can somebody help?
Post-translational modification.
Oh, post-translational modification, right.
No, no. I mean, the NMR spectra of the protein itself, specifically carbonic anhydrase, is really very the same in vitro that is produced by coli as well as in Excel. So then there are no modification of an amino acid, and really we detect all the amino acids. So except if there is acetylation of the first amino acid, this I cannot tell. But for the other is really the spectra superimposable.
2 more questions, quick. One is from Henry Hanneke. Thanks for the talk. Quick question, how do you deal with poor shimps resulting from inhomogeneous cell samples?
And we live with them. We live with them. Yes, there will be some broadening. Inhomogeneous broadening is present. There is, but we can. It's not a major problem. You see that the spectra are really good.
And then another from John Tainer. Once you have a working system, how long do these in-cell measurements take?
I mean, that is a matter of hours. I mean, if we do static, it takes 1 hour. They survive in the NMR tube, but they live for just an hour, 2 hours. And in the bioreactor, in the flow, they last as long as they want. But the spectra that can be acquired, again, depends on the concentration and the magnetic field, that they can be acquired in half an hour or so that it doesn't take much time.
Okay, we don't seem to have more questions. So I think we can thank Lucia, and I'll pass the floor to Marcus, I guess, again.
Stop the sharing.
Thank you, Lucia.
Thank you.
I guess my role is now to introduce Bernd. So Bernd studied physics and biochemistry. Actually, I did not know that or did not remember that at the University of Bayreuth. Then he did his diploma with Matthias Sprinzel and Markus Schwörer on NMR already of tRNA in E. coli. Then he did his PhD in my group on methods for analysis of constitution and conformation of macromolecules, natural products, and metalloorganic compounds. I remember that he was the first student, because my agenda was so full, whom I had to meet on the weekend. So he was able to and willing to meet, which was a great plus when he visited. Then he went on as a postdoc to Bob Griffin from 98 to 99, where he learned solid-state NMR spectroscopy and introduced actually at the time not so I mean, the solid state, I think, totally absent idea of deuteration in order to get better line widths for proton detection. So the first steps in that direction were done by him in Bob's group. And I remember that it took some effort to convince Bob that this was a good idea. Bernd can maybe comment on that during his talk. Then after 2 years, he went on to the Technical University of Munich as an Eminöter research fellow, which means that he was fully independent and could do his research. Then 3 years later, 2003 to 2010, he was a group leader for solid-state NMR at the FMP in Berlin-Buch, a colleague of Hartmut Oschkenat at the time, and also had then appointments with the first with the Charité and then also with the Humboldt University, and also was a coordinator of a graduate school there. And since 2010, he went back to the Technical University in Munich on a professorship, and is also has a role at the Helmholtz Center. And everybody who has a role at the Helmholtz Center has access to 2 sources of money. So that's always very beneficial if you have 2 hats on your head. So Bernd, please.
Okay, thanks a lot, Christian, for this very kind introduction. And also thanks, Markus, for the invitation to speak here so that I can share some recent results that we obtained in the group. And the title, as a title of the talk today, I've chosen Interaction of Small Molecules with A-beta amyloid fibrils by MIS solid-state NMR. And as you know, probably to date, there are more than 20 diseases known to be related to protein aggregation and deposition of protein material in various tissues. The most prominent one is presumably Alzheimer's disease, but also Parkinson's disease, where you have deposition of alpha-synuclein and Lewy bodies. Huntington's disease, but also type 2 diabetes is related to aggregation of proteins. And in this case, it's a hormone, IAPP or amylin, so human islet polypeptide, that deposits in the beta cells on pancreatic Langerhans islets. But for today, I would like to focus on the Alzheimer's disease A-beta peptide. A-beta is a cleavage product of the amyloid precursor protein, which is actually a type 1 membrane protein. Subsequent cleavage of 2 enzymes, beta secretase, produces a C-terminal fragment that is further on processed by gamma secretase that produces the A-beta peptide that subsequently aggregates into oligomers and fibrils that we would like to study. We use MIS solid-state NMR, that means magic angle spinning solid-state NMR. In this technique, the sample is rotated under an angle of 54.7 degrees with respect to the external magnetic field. And as a consequence, the static Pake pattern is split up into rotation sidebands. So these sidebands are separated by the rotation frequency. And when you spin faster and faster, you only retain the isotropic chemical shift that you're known from solution-state NMR. And to date, it's possible to rotate the sample with frequencies up to 100 kilohertz if you use a 0.7 millimeter rotor. The volumes become really small, but we have to make sure that the surface velocity does not exceed speed of sound. The amyloid fibrils, they are characterized by typical features, and as the name says, they are fibrillar structures as seen here in the electron micrograph. And by X-ray diffraction, this basically resulted in the name cross beta, because you have 2 reflections, one reflection at a scattering of 4.7 angstroms. This corresponds to the distance of peptides that are hydrogen bonded along the fibril axis, and another scattering at a distance of 10.7 angstroms. And this corresponds to the distance of the amide bonds that are separated by a beta arc in the plane of a fibril. Basically, all amyloid fibrils have beta sheet propensity, as seen here in CD spectroscopy, and they're characterized that they are THT fluorescent. Thioflavin T is a small molecule that consists of this dimethyl aniline ring and this dimethyl benzothiazole moiety. And this bond has a single bond character, so the molecule is freely rotatable. And as a consequence, in the non-bound state, the fluorescence is quenched. And in the bound state, the rotation is hindered, and this induces a high fluorescent quantum yield because of the immobilization of the ring. It is thought that these dyes, it can be thioflavine T, but Congo red is another one, they bind along surface side chain grooves in parallel to the long axis of the fibril. And here's an example from Gerd Meyer's group, so Congo red binding to head S fibrils. And you can nicely see here how the Congo red binds to the surface groove. And I should say that this binding is very unspecific. So it's basically used as a definition for interaction with fibrils. And thioflavine T binds independent of the kind of molecule. It can be A beta, it can be alpha-synuclein, it can be tau. They all recognize an amyloid fibril. And even though that it is rather unselective, these small molecules are actually used for imaging, for PET imaging. And PET represents positron emission tomography, in which you label your small molecule with a radioactive nucleus. So you see this is the original thioflavine T molecule, and the Pittsburgh compound B, that is represented down here, has a very similar scaffold. Again, you have this methyl aniline moiety and the hydroxybenzyl thiazole moiety here on the left. When a radioactive nucleus, if it decays, then this induces an annihilation event. So the positron hits some organic matter, then this causes annihilation, and this means emission of 2 photons or 2 gamma quanta in opposite direction. And from runtime differences between runtime differences between these 2 photons, you can localize and the position of the radioactive nucleus in the specimen. In combination with CT scanning, you can then find out where you have plugs in the brain of the patient. So these THD-derived molecules is one class. Another molecule like Vizamyl that is marketed by GE is basically flutemetamol. These 3 molecules are based on the thioflavine T fold. In FIB-T, you have an imidazole moiety linked to the benzothiazole moiety. And in the fluorbetabine, which is marketed by Bayer under the name Neuraceq, you have the stilbene moiety to image plugs. And as I said before, so these molecules are all unspecific, and this is obviously not good when you want to differentiate between different neurodegenerative diseases, and you aim for molecules that either recognize Alzheimer's disease A beta or alpha-synuclein in Parkinson's disease. And Hans-Jürgen Wester here at the TUM in Munich, they developed molecules that have these bithiazole, diaryl bithiazole scaffolds that have this selectivity. And if I try to walk you through the table, there's, for example, compound 1 that has a high affinity to A beta, but basically no affinity with respect to tau and alpha-synuclein. And by contrast, compound number 2 has a high affinity to alpha-synuclein, but no affinity to tau, and a relatively low affinity to Alzheimer's disease. And compound number 3 finally has high affinity to all 3 classes of these molecules. And it is basically the aim of our animal studies to find out what is the molecular mechanism of binding of these small molecules to these amyloid fibrils. And what you see also here on the table is that the C-log p-value for all of these compounds is very high. So the C-log p-value is basically the ratio of the solubility or the partitioning coefficient of the solubility in hexanol or octanol versus the solubility in water. And this means that these molecules are very well soluble in octanol, but basically not soluble at all in water. And this means that this poses some problems for our NMR titration experiments. So I would like to show you now the titration protocol that we employ to titrate these hydrophobic small molecules. As I said, so the molecules are very hydrophobic and basically not soluble in water. Normally what you do when you do screening in solution state NMR, that you have a library of compounds that is dissolved at high concentration in DMSO, and then you dilute this into your aqueous buffer in the protein, and you have a final concentration of DMSO maybe on the order of 1 to 3%. And this is how you do drug screening. However, if you do this with these A-beta fibrils, then we see that actually DMSO, even if it's only added at a concentration of 1%, it perturbs the fiber structure. So in black, you can see here a PDSD carbon-carbon correlation spectrum for the A-beta reference structure. And I only show you here the part of the spectrum that contains serine cross peaks. And when we add 1% DMSO to the sample, then you see that new peaks come out and the old peak actually disappear, which would indicate that DMSO, even this low concentration of DMSO, perturbs the fiber structure. That's also expected maybe because DMSO presumably binds there. And we thought of ways, how can we overcome this issue? And we were wondering if we can take bovine serum albumin as a carrier system to transfer our small organic molecules to the A-beta fibers. And as you know, human serum albumin, for example, is also used in the plasma to transport organic or non-soluble molecules. And we are wondering if we can also use this as a carrier for the DAPTAs or for the PIP molecules. And let me now show you the titration protocol in more detail. So BSA is a very nice protein because it's very forgiving with respect to DMSO. So the protein is still folded in a concentration of 25 volume % DMSO. And this is a condition where the organic molecules, the DAPTA molecules, for example, are soluble. And I should say also DAPTA, in this case I use DAPTA because it has yellow color. So we can follow the titration protocol and the whereabouts of our molecules basically with a naked eye without having to use a spectrophotometer. But the protocol works the same for PIP or others. So the first step is the solubilization in a water DMSO mixture. Then next we have a washing step to get rid of the DMSO. So we use a centricon or an armicon and wash the DMSO out. And then finally we add our amyloid fibrils. So we have this this yellowish-like solution. And now the next step consists of separation of the DAPTA-bound fibrils and the BSA. And this we achieve by just simple sedimentation. So we centrifuge the solution, and you see here that actually all the DAPTA-bound fibrils are going into the pellet, whereas basically the supernatant doesn't contain any DAPTA molecules or DAPTA-bound BSA any longer. And this we confirm always also photometrically that we see that DAPTA binds quantitatively to the fibrils and nothing is left in solution. So what are the advantages of this titration protocol? So as I mentioned before, the first advantage is obviously that there is no DMSO in the sample anymore. The second advantage is that we can now really do concentration dependence of the ligand. We can prepare the ligand-bound sample. We measure solid-state NMR spectra. We unpack the sample, wash with higher concentrations of DAPTA, and pack the rotor again. And as you see here in the carbon 1D spectra, also nitrogen 1D spectra, we basically lose maybe on the order of 5 to 10% per washing step. So this is still acceptable. It means that we are very conservative with respect to sample, but what is even a greater advantage is that we can always work with the same fibril conformer, and because reproducibility is sometimes an issue, and in this way we make sure that we always have the same fibril conformer in our solid-state NMR rotor. And obviously the goal is to identify the ligand binding site in the amyloid fibril. And this I would like to show you in the following. So this shows a 2D PDSD carbon-carbon correlation in black for the A-beta fibril reference, and in red for the sample that is washed with a 4-fold molar excess of the DAPTA molecule. And when you zoom into this aliphatic part of the spectrum, then we're very disappointed because there were virtually no chemical shift changes. So you can see that the spectra are highly reproducible, but no shift changes. And this was a bit disappointing. And we're wondering what are the reasons why we don't see any shift changes. And the first possibility is obviously that the molecules bind to the region of the A-beta fibrils that are not assigned. As you probably recall, A-beta fibrils are not fully structured from the N to the C terminus, but we can assign here the core of the fibril, basically consisting of the second half of the peptide, whereas there's only very few assignments here in the N-terminal half. So this is one possibility. And another possibility could be that actually we are detecting carbons. Carbon have a low gamma, and due to the evolution of carbon-carbon J couplings in both dimensions, we have very broad carbon resonances. So they're typically 150 Hertz broad or even broader. And this could make an identification or small chemical shift changes difficult. Okay, and just for comparison, I would like to show you a different system that we worked on in the past to also show that this result is was unexpected, again, given the fact that we have quantitative binding of these small molecules to our amyloid fibers. And this is sulindac sulfide. Sulindac sulfide is a non-steroidal anti-inflammatory drug that we investigated together with Gerd Multhaupt's group at McGill University in Montreal. And that was shown to bind to A-beta fibrils, but also reduce cytotoxicity. So here are A-beta fibrils incubated together with cells in the absence and the presence of the small molecule, and you see that cytotoxicity is reduced. And what we observed there is actually very large spectral changes upon titration of an excess of sulindac sulfide to A-beta fibrils. And what you see, for example, here for alanine 21 or also for leucine 19, that the original resonance disappeared totally in the presence of sulindac sulfide. And that would indicate that for each and every fibril, for each and every peptide in the fibril, we have binding of sulindac sulfide to the peptide. And in this way, we could identify the small molecule binding site for sulindac sulfide. But again, for PIP, we don't see these very large chemical shift changes. So we thought maybe this is the reason could be that we are having, the carbons have a very low gamma, and the binding maybe is more subtle. And therefore, we turned to deuterated samples and recorded proton-detected experiments. In particular, we looked at HCA NH experiments and HCA CONH type experiments to get sequential assignments. And as you see again, we can assign the C-terminal half of the peptide. I show you the spectra that we obtained in two flavors. So on one hand side, we have these proton-nitrogen correlation spectra that you're used to. But in a second, you will also see that it makes sense to represent the backbone as an amide proton to C-alpha carbon correlation experiment. And then you have a closer look at these spectra, then you see that some of these resonances or some of the residues come in two sets of resonances. So for example, here glycine 33 is indicated. We not only have this cross peak, but a second one. And as you see that actually this cross peak seems to be smeared out. We cannot sequentially assign this peak here, but it seems also to belong to glycine 33. And the same is true for other glycine residues, glycine 25, serine 26, alanine 30, isoleucine 31. Now, what happens when we titrate a PIP molecule, so Pittsburgh compound B, to these perdurated fibrils? So this shows the A-beta reference spectrum here in the presence of the C13 labeled PIP molecule, and here the overlay. Then you see that for most of the backbone amides, not much is happening. So the chemical shifts are basically unperturbed, but you see that there are large chemical shift changes here in the C-alpha region. Again, this is the amide proton to the C-alpha carbon correlation experiment. And if we now focus on one hand side in to residues for which we find two sets of peaks. And on the other hand, if we focus into the glycine spectral region, then we end up here. Then we see that actually in the presence of the small molecule, the structural heterogeneity that we observed is actually abolished. We only find one set of resonance in presence of PIP. At the same time, we find large chemical shift changes, in particular for residues glycine 25 and glycine 33. So the glycine 25 peak walks from here to here, and the glycine 33 peak goes from here to here, whereas other peaks are not affected. And at the same time, also for glycine 37, you see that this structural heterogeneity that seems to be present in the reference spectrum is not there anymore. So again, chemical shift changes are mostly localized in this loop involving the JSMK loop involving residues 25 to 28, and it is also localized around residue glycine 33. Now we are wondering how we could represent the ligand binding in kind of a structural way. So we started out with a structural model that has been put forward by Rob Tico's group already quite some time ago with Ani Petkova. And this consists of this twofold symmetric dimer structure. And when you have a closer look into the structure, then you see that there are cavities in the structure, so that regions of the structure that are not filled with density that can potentially accommodate a hydrophobic small molecule. And one of these cavities is located, in fact, between residues 25 to 28. And another cavity that is even larger is located on one hand side around residue glycine 33. On the other hand, we have here phenylalanine 19. Now you might argue that these cavities are due to the fact that the structure maybe is not very well defined, is not compacted, because there was, at that time, there were not enough distance restraints to determine the structure. And therefore, I looked up a few other cryo-EM structures that have been determined recently. So here on top, I show you the dimer structure that has been calculated and determined by Markus Fendrich's group. And here is the A-beta 42 structure that has been determined by Gunnar Schröder's group. And also there, and here I have drawn spheres into the structure where there are cavities that have at least a diameter of 5 angstroms. So they would be large enough to accommodate a hydrophobic molecule. And as you see, and this is maybe also clear, wherever you have glycines in the sequence, the glycines lacking a side chain, and here you have particularly a lot of space to accommodate a small molecule. Okay, so next we wanted to actually probe direct interactions between the small molecule and the A-beta fibril, and this is also the purpose or the reason why we labeled the PIP molecule here with C13. So we have here two methyl groups attached to the aniline ring, and we have methylated the hydroxyl group that was originally there in this benzothiazole ring. And when we recorded the solid state in the mass spectrum, we were very disappointed because the N-methyl proton chemical shift would be expected to be around 4 ppm, the same for the O-methyl proton chemical shift, and the carbon shifts would be expected in the range between 40 and 60 ppm. However, when we look into the solid state NMR spectrum, there's basically no cross peak. And potential reasons for that is, on one hand side, it could be that the ligand goes in and out of the binding site, and we have some exchange broadening. I think we can exclude this possibility because we use a fourfold excess of the ligand. And even if there would be like 3 binding sites, or it's expected that there are 3 binding sites of thioplasmin T to A-beta fibrils, under these conditions, we would actually not expect a large amount of chemical exchange broadening. And another possibility is actually dipolar line broadening, because we carried out this experiment all in a 1.3 millimeter rotor, and means that we employed an MAS rotation frequency of 55 kilohertz. And in order to appreciate the effect of dipolar line broadening, I I would like to show you a few simulations, Simpson simulations that we carried out in collaboration with Sternig-Toschner's group. In these simulations, we assumed a per-deuterated alpha-spectrine SH3 sample, in which certain methyl groups from valine and leucine had been incorporated stereoselectively. So the system is very proton dilute, only a few methyl groups are protonated, and then we determined or calculated the expected intensity of these methyl proton resonances. And you can see that for few like leucine 8 in the system, we need like 100 kilohertz of MAS to achieve approximately 80% of the maximum signal intensity. When we do the same, or if we would look at another residue like valine 9, then we would see that we only achieved like 30% of the maximum intensity. And situations obviously much worse if we would be at 50 kilohertz. We also compared these simulations with experimental data, and this is shown on this slide. And we did, for SH3 at least, we carried out solid state NMR experiments in the frequency range between 50 kilohertz and 110 kilohertz. And you see that the experiments match actually rather well with the simulations. Experiments are in red and the simulations are in black, except maybe for leucine 8. This is the most N-terminal residue that we can see in the structure, and there we expect a few crystal contacts that have not been taken into account in the simulation. And what this tells us that actually faster spinning probes are necessary to look at these methyl groups. So even if we are working in a very heavily deuterated environment, and the rotation frequency that is necessary to achieve 50% of the maximum sensitivity would be on the order of 135 kilohertz on average, depending again on the proton spin network. So this brings me to the end. I hope I could show you that BSA can be employed as a carrier system to titrate small hydrophobic molecules. We observe very subtle chemical shift changes, and these involve mostly glycine residues that are located in turn regions of the A-beta fibril structure. We can see these shift changes only in proton-detected experiments. In the future, we would like to repeat the experiments at faster rotation frequencies. And in particular, we also would like to look at different polymorphous form of the A-beta fibrils. In the lab, we are already able to reproducibly produce different morphs. So you can see this here in the serine region of the spectrum. The pattern of the serine C-alpha C-beta cross peaks is distinct, and there's only very little, so to speak, impurity from the second polymorph. And we would like to know if the binding is different if we look at different polymorphous form of the A-beta fiber. So I would like to stop here and thank the people who were involved in the work. The project that I presented on these PET tracer molecules was one PhD project from Chong Yu, when she was a PhD student in the group. The Simpson simulations had been done by Sternig-Toschner together with Kai Xu, and all the NMR experiments have been supervised and assisted by Rediman Sarkar. The new experiments are done by Natasha Rodina, and the sulindac sulfide experiments were done by Elke Prade. And this was a collaboration with the TUM and the Universitätsklinikum Marburg with Berus Josefi and Hans-Jürgen Wester. And I would like to thank you for your attention.
Yeah, thank you very much, Bernd, for this beautiful talk. And I start with one question, which is also my question from the questions here from Henrike Heise. If you add the binding molecule to mature fibers, do you think it is realistic that the organic molecule can still enter into the internal cavities of the fiber?
Well, that's what we see. But at least it binds strongly. So we start with preformed fibrils. We wash it. And well, with this color assay, we see that it's bound. The question is if it would rather bind at the inside or on the surface.
I guess that it is somehow for, I mean, if one also compares with other structures from binding molecules, everything so far has bound on the outside. So it's a valid question.
Yeah. But I would compare this a little bit to membrane protein binding, right? So if you follow this argument, then there would be no reason why a membrane protein or a membrane penetrating peptide would ever bind within the membrane, and it could only bind at the surface because there's no space. But I think we believe that it binds in the interior because there it's more hydrophobic. On the surface of the fibril, one phase of the peptide would be still to the solvent, and I guess the small molecule doesn't like this. So I think it's more happy if it's really embedded in a hydrophobic core.
Do you have any idea, bypassing the questions, but then I read the questions. So any idea about the stoichiometry?
That's a good question. There are people doing equilibrium dialysis experiment, and they identify multiple binding sites. So I think, well, obviously the stoichiometry for a particular binding site is a 1-to-1 stoichiometry, but there are at least 3 binding sites known.
Okay, now I start with real questions here. John Tainer, beautiful results for the BSA protocol to transfer from DMSO to amyloid target. What is your ideal working ratio of BSA to dye-ligand molecule? Do the glycine results give you clues about fiber formation mechanism?
I think in the BSA titration protocols, we always have used a little bit of an excess of the BSA to make sure that all the small molecules is binding. But I think also there, it's known that BSA has multiple small molecule binding sites. So I think we could even use the BSA substoichiometric. But we didn't try this. So because solubility was not an issue, and that's why we approximately had a 1-to-1 stoichiometry. The second question, I forgot.
It's now gone. I also forgot. Wait a second.
The glycine results give you clues about fiber formation mechanisms. No, that would be interesting.
Yeah, exactly. Yeah,
interesting point. But we don't understand how glycines are related to seeding and if they can trigger one polymorph or structuring from one polymorph into another. No, we don't know.
Okay. Simon Olson asks, Bernd, nice talk. Is it known whether the small molecules stabilize the fibril state? Can the chemical shift perturbation tell us something about this?
Actually, we wanted to do, that's a very good point. We wanted to do these experiments to test the thermodynamic stability of the fibril in presence and absence of the small molecule. But I have to apologize, we didn't do it yet. But I would expect that actually the small molecules would increase stability. Okay, then Patrick van der Velde about the CH3 simulations. Did you explicitly include dynamics of the CH3 in those calculations? Is the MAS dependence due to interference between mobility and the MAS? So Patrick refers to the SH3 results, right? Yeah, CH3. Mm-hmm. And what we didn't mention this, but we included dynamics there, and then think there was, maybe you can go back in the slides if this. Yeah, you can. So you have here the first line, we just assume a rigid order parameter or rigid limit order parameters for the methyl groups. And in the second line, we have measured side chain order parameters with Redo type experiments, and they obviously also scale down proton-proton dipole interactions, and we employed this for these calculations, and there we ended up with a smaller value for the rotation frequency that is needed to achieve 50% of the maximum possible sensitivity. Yeah, this is accounted for. Then an anonymous attendee says, how did you determine the error bars in the chemical shift perturbation plots? No, we basically used the line widths, information on the line width of the resonance, and this would reflect then the error bar in the CSP. Okay, I mean, we will have the informal session afterwards. So if you are not satisfied with the question, of course, everybody is invited then finally to raise the hand and
and ask and grill Bernd and of course Lucia as well afterwards. Ricarda Turner, the DMSO has been reported to be a chemical chaperone. Maybe it would be interesting to investigate the fibril binding a bit further. Do you think there could be a disaggregation process taking place?
Well, this low concentration, I don't think that disaggregation really takes place. If we would increase the DMSO concentration, I also believe that this would change the equilibrium between the fibril state and the monomeric state. And we didn't do this so far because DMSO doesn't really occur in nature. And that's why we didn't want to touch on DMSO so much.
Good. Thomas Schmidt, could CEST or DEST on the small molecule measurement create some understanding on the binding modes?
Yeah, that's a good point. We thought about this, but then on the other hand, when you remember how we prepare our fibril samples, we sediment this, and we believe that the binding is that strong that there is actually no PIP or adapter molecules free in solution. So we do not have this excess ligand because we just purify it away, and we can only have maybe exchange within the fibril between these different binding states, and this is something that I think should pursue to really quantify the chemical exchange process. So I believe that there is chemical exchange between the different binding sites, but we didn't try anything yet to quantify this.
Then Rams congratulates you to an excellent talk. Then he asks, did you observe THT binding to be disrupting the amyloid fiber structure of A-beta, or did you observe more than one polymorph induced by THT binding?
Well, as I said, so well, we didn't use THT. We used this PIP molecule or adapter molecule, which is very similar to THT. But in the first place, in the carbon detected experiments, we didn't see any effect whatsoever on the spectra. Only in the proton detected experiments, we saw some small effects. And I don't believe that we see a disruption of the fiber structure.
Mm-hmm. Okay. Then here I have a question by Philipp Neudecker. Bernd, did I see that correctly, that you have glycine carbon C alpha at 38 ppm? Is there a rationale for that it is at this chemical shift?
It's he probably was referring, or Philip, you're probably referring to glycine 29. No, I don't have a good reason why this shift is so much upfield shifted.
Okay, maybe.
But it's actually, this residue is in this turn, in this hypothetical turn. So it could be that it maybe is not a regular residue in the Ramachandran diagram. Don't know.
Okay, then Hartmut is following up on Patrick's question on the CH3 results and asked whether you tried to measure at very low temperature.
No, we didn't try. Well, Hartmut knows that this, but this is for the A-beta or the SH3. So for both, we didn't really measure at low temperature. For okay. A-beta, that might be actually interesting to suppress exchange.
Elliot Crooks is asking, a recent cryo-EM model of vascular A-beta fibrils, Fendrich 2019, suggests a loop in residues 1 to 28, while residues 29 to 40 are solvent exposed. Could this explain why binding and loss of heterogeneity occurs in the C-terminal region?
Well, I have to admit that we did not determine the fiber structure yet. We always wanted to do this, but we still don't have a 0.7 millimeter probe. And with a deuterated sample, it turned out to be difficult to do that. But obviously, one big issue is to know which polymorph we're actually working with. So we're working on the fiber structure, and then I hope we know better.
Mm-hmm. Okay. Umut Günzel, I guess the flexible N-terminal part of the A-beta peptide is more hydrophilic. Would it be possible to load the dye into the peptide without the C-terminal part from BSA? If the peptide dye complex is soluble, maybe one can use solution NMR.
Yes, but I understand it like this, that the thioflavin T recognizes an amyloidogenic core. If I only have the N-terminus, I suppose that it doesn't bind. And
Makes sense. Manda Bobardika is asking, interesting work. Could you please explain the reason for appearance of multiple peaks for G33 and the nearby residues?
yes, we assume that actually the protein can adopt different conformations in this region, and maybe switches back and forth between different polymorphous forms. And that would also make sense because we hypothesized that we have these cavities. That means that the side chains or the residues around there have certain flexibility. And if the ligand binds in the core, then the side chain is pushed in one or the other conformation, and these multiple conformations are not possible anymore.
Ipin Agarwal asked, thanks Bernd for the nice talk. I missed the point how PIP induces the heterogeneous fibrils into a homogeneous sample, at least in terms of chemical shifts.
It seems to me that this is a very similar question as I tried to address right now, that these regions where we have these cavities can exist in multiple conformations, and when the molecule is present, then there is just no space for this exchange process anymore.
Myung-Kyu Kim is asking, thanks for the nice talk. Did you check the concentration of the small molecule in the supernatant after collecting fibrils?
0. Everything's gone.
It's also due to the fact that the solubility is so low, right?
Well, the BSA is still present.
The BSA is there, yeah. Ekaterina Burakova, thank you for the talk, Bernd. Have you recorded any dynamics on A-beta? Do you know if one amyloid form, one set of peaks, is more labile than the other?
That's a very good question. I have to admit that we didn't do this yet, but we are starting, well, not with this system, with IAPP.
Jiafei Mao, will you give shot for 19F containing compounds? Maybe better chance on getting restraints.
We did this with a sulindax sulfide. So sulindax sulfide has one fluorine in the core, and we measured distances between sulindax sulfide and the amyloid. But the PIP molecules are not fluorinated. Well, they can be fluorinated, like for PET imaging with F18. So I guess one can also introduce an F19 nucleus, but we didn't do this yet.
Venus, me too, is asking whether the log P of your PET compounds is not too high.
Actually, well, it's used in vivo, right? So the molecules are used in vivo. We know that they are working. And the good thing for PET is that one doesn't need very high concentration. So when you inject it in a nanomolar range into the body, this is sufficient, and it's soluble on this, in this order. So for PET imaging, this is not an issue.
Stefan Becker asked, are there any plans to check the structures of the compound-bound fibers by cryo-EM?
I think people are doing this. Actually, a few days ago, I've seen a paper from Sorcheres and the Cambridge people on PET tracer molecules binding to tau in after neuropathology. But we didn't try this yet. I always, well, obviously, I'm doing NMR and I believe in NMR. And I think it's difficult to analyze the ligand binding site with cryo-EM because it's known that these PET tracer molecules can bind in different sites. And this is actually what we are interested in. We would like to characterize the exchange between these different sites. And I would expect that due to that, cryo-EM probably yields always relatively low resolution.